progressing ex vivo gene editing innovations with high-quality Leukopaks. Being able to treat human disease at the genetic level has been a long-standing goal, yet it wasn't until 2012 that momentum surged. The discovery of the CRISPR-Cas9 gene editing system gave researchers the unprecedented ability to precisely target and modify genes with much greater ease than first-generation genome editors. In late 2023, gene-edited medicine reached a significant milestone with the approval of Casgevy, a CRISPR-edited gene therapy to treat sickle cell disease and transfusion-dependent beta thalassemia, ushering in a new era of genomic medicine. Ex vivo gene editing has been used to successfully develop hematopoietic stem and progenitor cell-based gene therapies to treat a range of diseases. More recently, gene editing is being used to enhance the safety, efficacy, and in vivo persistence of CAR T cells, as well as its role in the development of universal CAR T therapies. Leading biopharma companies are developing ex vivo hematopoietic stem and progenitor cell-based gene therapies to treat blood-specific disorders such as primary immunodeficiencies, as well as other disorders including Gaucher disease and Hunter syndrome. Because the normal circulating percentage of CD34 cells is relatively low, mobilization is used to increase their numbers prior to leukapheresis collection. Single or dual agent mobilization regimens using G-CSF and plerixafor can effectively stimulate the release of CD34 cells from the bone marrow into the bloodstream. Studies have shown that dual mobilization significantly enhances HSPC yields compared to either agent alone. As you can see, using ISAGE protocol, the percentage of CD34 cells from Discovery's all-cells dual mobilized Leukopaks is 1.29% for the G-CSF and plerixafor regimen, compared to single agent mobilization using G-CSF at 1.07% and plerixafor only at 0.43%. High-yield, high-quality CD34 HSPCs from a single donor are essential for efficient genetic manipulation in mobilized Leukopaks. The problem, though, is that mobilization agents like G-CSF not only mobilize stem cells, but also stimulate granulocyte production, increasing their numbers within a Leukopak. While some granulocyte presence is expected, excessive contamination can compromise CD34 cell quality, impacting cell viability and functionality. Controlling granulocyte levels is essential to prevent quality-related issues that affect downstream gene editing. Thanks to optimized protocols and decades of expertise, Discovery's all-cells mobilized Leukopaks maintain the lowest content in the industry. As shown, on average, the granulocyte content is 3.3% in a non-mobilized Leukopak. When compared to a mobilized Leukopak collection, that percentage increases to 8.86% for dual G-CSF plerixafor and 9.29% for G-CSF alone. CAR T cell therapy has transformed cancer immunotherapy, but several hurdles limit its widespread adoption as a first-line treatment. Gene editing technologies are being used to address key limitations, including improving CAR T safety by reducing off-target toxicities, enhancing in vivo efficacy by improving CAR T survival, metabolism, and responsiveness to tumor signals, enabling the development of universal CAR T therapies from donor cells by reducing the risk of immune rejection and graft versus host disease. Gene editing supports allogeneic CAR T strategies to broaden patient access and reduce manufacturing costs. For CRISPR-Cas9 gene editing of CAR T cells, the target cells need to be highly viable and in active phases of the cell cycle to promote efficient homology-directed repair. Therefore, the viability of the CAR T cells, which is largely influenced by the quality of the starting T cell population in the Leukopak, directly impacts the success of the gene editing process. Throughout our Leukopak application series, we have highlighted different use cases for both non-mobilized and mobilized Leukopaks in the development of next-generation therapeutics. These discussions highlight the critical role of high-quality Leukopaks, where both donor and process-dependent variables are optimized for success. As cell and gene therapy continues to advance, donor-derived starting materials will be an essential starting point to drive innovation and deliver life-changing treatments to patients.